Hello, and welcome to the DDW Cell and Gene Therapy Showcase. I'm Rhys Armstrong, the editor for Drug Discovery World. During today's showcase, which is being co-sponsored by Benchling, Sartorius, Applied Stem Cell, and Elegant Bio, leading industry figures will outline areas of opportunity and challenges in the cell and gene therapy, drug discovery and development sector. The showcase will also include a panel discussion exploring the global market for cell and gene therapy development, as well as the tools, concepts, and technologies driving forward research in this area. We will start the showcase with an expert panel discussion consisting of Jacqueline Barry from Cell and Gene Therapy Catapult, Laura Campana from Resolution Therapeutics, and Dr. Neil Flomenberg from Tavogen, as well as our sponsor speakers, Galit Meshulam-Simon from Elegant Bio, Tim Largan from Applied Stem Cell, and Hayley Merritt from Benchling. I'll now hand over to DDW Multimedia Editor Megan Thomas and the panelists.
Hello everyone, and welcome to the panel discussion of the Cell and Gene Therapy Showcase. My name's Megan Thomas and I'm the Multimedia Editor at DDW and the host for today's discussion. I'd like to start by going around the panel and asking everyone to introduce themselves, what they're working on, and to share where you're based and whether you think that is a global hotspot for research funding and clinical trials in the CGT space. So Neil, could we start with you?
Good morning. I'm Neil Flomenberg. I'm the Chief Science Officer for Tavogen Bio. Tavogen is interested in cytotoxic T cells and how this tool of nature can be utilized in ways to help deal with infectious diseases, infection-triggered cancers, and sporadic cancers thereafter. I think that there are lots of opportunities for funding. It's hard to pick and choose, so I would say that we probably feel we are in a hot spot.
Great, thanks Neil. Galit, can I pass over to you?
Yes, sure. Hi, everybody. My name is Galit Meshulam-Simon. I'm Associate Director of Commercial Applications at Elegen. Elegen is a company which is based in San Francisco Bay Area, and we have a novel next-generation technology for DNA and gene synthesis, which is basically passing using cells, so we are completely cell-free. With that, we are providing DNA, working with many clients and collaborators about expediting the cycle of using DNA and helping with cell and gene therapy. We think that, yes, the Bay Area is really prime location and the hotspot for cell and gene therapy. There is a unique combination of academia. We have, I would say, world-known research institutes like Stanford, Berkeley, UCSF on one hand that are working on cutting-edge technology. They're also contributing a lot of talent pool to this area. There are many established biopharma companies like Gilead, Genentech, and the combination of that with a very collaborative ecosystem and a place where is also attraction for many venture capitalist money and grants, governmental grants, is really making it a prime location for cell and gene therapy research. And I think that's true for research and commercialization as well.
Thank you for that. Tim, would you like to go next?
Yeah, I'm Tim Largan. I am with Applied Stem Cell Incorporated. We are a CRO and CDMO in the cell and gene therapy space, supporting researchers and clinical development of companies that contract with us. We are located in the Bay Area of California, so technically in Milpitas, which is on the South Bay near San Jose. And it is definitely a hub for funding and clinical trial activity and R&D in particular. Ranks up there with Boston kind of every year. It's in the top 2 or 3.
Yeah, a busy area, and it does seem like everything's going very global. Not everything is necessarily needs to be within those kind of golden triangle original areas. Can I now hear an answer from you, Jacqueline?
Hi, thanks. I'm Jacqueline Barry. I'm the Chief Clinical Officer of the Cell and Gene Therapy Catapult here in the UK. We have a number of sites in the UK. We have them in London, Braintree, Stevenage and Edinburgh. And at the Cell and Gene Therapy Catapult, we're really committed to advancing cell and gene therapies with a vision of bringing a thriving industry delivering these life-changing therapies to the world, essentially. And we do that in a number of ways. We're a team of experts covering many, many disciplines, and we work with developers and the healthcare system to develop the technologies and then to drive the adoption of this innovation into healthcare. In terms of, do I think the UK is a hotspot? I absolutely do. I think we've got many advantages here in the UK. For research, we've obviously got some, world-leading institutions in this space, such as Francis Crick, or indeed the Edinburgh University, where I'm sat just now. There's many, many scientists who are involved with spin-outs from a number of universities. And Lara, who's also going to be on the webinar today, will talk about one of them, Resolution Therapeutics, which is from Edinburgh. But there's many other examples: Crucible Therapeutics from Sheffield, Orchard Therapeutics from UCL, MatchBio from Oxford. So I could go on, there's many, hundreds of them. But also, it's very rich for funding. The collaborators here in the UK of Cell and Gene Therapy Catapult raised over £600 million last year. And the BIA have surveyed and seen that UK companies secured about 55% of all the European VC funding. And in terms of clinical trials, we have representation about 9% of all global clinical trials for advanced therapies, with a really strong pipeline coming. So I think I can safely say that I think the UK is quite a hotspot for advanced therapies.
Great, thanks so much. Hayley, can we move on to you, please?
Yeah, hi everybody, I'm Hayley Merritt. I'm a scientific solutions consultant here at Benchling, and we work with teams looking to get on a unified platform and just find better ways to manage their data. And I'm based on Long Island in New York, so about an hour east of New York City. So thinking about if we're a hotspot, I think New York, there's probably funding and many companies have a presence in New York City. But I'm also not too far from some of the more research-focused areas in New Jersey or Philadelphia. And then I'm also actually only 20 minutes away from Cold Spring or Harbor Labs on Long Island. So a really critical center in kind of the history of DNA research, and continues to be an important research center today.
Great, thanks so much. And now, Lara, can we move on to you, please?
Thank you, Megan. So I'm Lara. I'm the SVP for Research and Translational Science here at Resolution Therapeutics. We are an Edinburgh and London-based biotech company. We're a spin-out of the University of Edinburgh. We develop macrophage cell therapy, and in particular, we pioneered the utilization of regenerative macrophages to tackle inflammatory and fibrotic diseases, with our first indication being decompensated liver cirrhosis, which is a large and growing unmet medical need. I'm in the same ecosystem as Jackie, literally just on the other side of the road at the moment. So yes, definitely second everything that Jackie has very eloquently said. And maybe just adding that Waikato per se may not be historically be considered a hotspot, but we are certainly one of the largest and fastest growing hubs outside of London and the Golden Triangle in the southeast. So definitely a nice place where to be to bring upon some innovations, some advanced therapies.
Great, thanks so much. And so that's actually quite a natural point to ask Jackie if you could elaborate a little more on some specific advantages for cell and gene therapy, the cell and gene therapy industry in the UK.
Thanks, Megan. So I suppose the UK is, geographically we're quite a small area, but we have many, many developers within that area. So there's a real network formed within the UK, and we've got the Cell and Gene Therapy Catapult. We have 2 manufacturing facilities where our collaborators can work with us to develop the products in a safe space and then move out into their own manufacturing plant, or indeed move to a CDMO, of which we have many, including, if I stick with the theme of Edinburgh just now, we have Roslin CT just beside us, who were the first manufacturer for Vertex product, first CRISPR gene-edited product. We also have a very permissive regulatory environment. The MHRA actually was the first global regulator to approve the Vertex gene therapy product, Casgevy, for sickle cell. And we also have a policy-driven approach. You know, the UK government takes a policy-driven approach where we look at the ecosystem as a whole, looking at how do we improve clinical trials, how do we upskill the workforce, how do we help developers move their product along, and they've invested in cell and gene therapy catapult. And indeed, we have investment also in something which I'm responsible for the coordination of. It's called the Advanced Therapy Treatment Centre Network, where we work with academia, industry and the healthcare provider in the UK, the NHS, to create a permissive environment to allow the acceleration of the adoption and or clinical trial of these therapies. So I think that holistic approach really does give the UK quite an advantage for the development of these products.
Great, thanks so much for that answer, Jackie. Gali, I was hoping now we could hone in a little bit on workflow, and I wanted to ask what the practical meaning of the 1 to 70,000 BP error rate is and how that affects researchers' workflow.
Thanks, Megan. So I will answer that. That's a technical question, so I'll explain a little bit more. So Elogen is a company that is making synthetic DNA. We are very proud of our very low error rate, which is really low compared with other linear DNA providers. So the meaning is that in average, when you're talking about such low error rate, it means that in average 99.999 of the molecules that we are providing will be sequence perfect. And why is it important? Important because it saves time and money for the clients later on with their DNA applications. In this level of accuracy and the fact that every molecule, every gene that we ship is passing our QC spec, which is over 80% of it will be full-length genes, it means that 95% of the portion of molecules that we are sending is sequence perfect. So if you take that DNA and put it into your application, for example, you're doing another cloning and you're picking colonies, all you have to do is to pick 1 to 2 colonies to get the sequence perfect genes. And of course, if you're using linear fragment, you don't even have to do that. But that means that it really saves time and cost down the line for our clients that are working with this DNA.
Great, thanks for that answer. Now, Lara, I was hoping we could then move to a slightly more general question next for you, which is what are some of the primary barriers in CGT research and how can these be tackled?
Yes, so I think one of the barriers that at least we have encountered, especially at the very beginning, and I need to caveat that we started the company during the pandemic, so that could also have been part of it. But definitely, I would say at the beginning, we really struggled to find the correct skill set, especially when it comes to the process and analytical development of the cell therapy. Obviously, the Catapult is actually doing a great work in the United Kingdom and I won't steal Jackie's thunder, but they have a fantastic training program trying to bridge that gap. But that's definitely something that needs to be worked as the cell and gene therapy space grows. The second thing I would say is that we have at least found is that there are some parts of clinical practice that are very well versed in cell therapy because they have had years of experience from the CAR T development or from gene therapy development, such as Casgevy. So what happens though is that if you branch into fields of medicine where ATMPs have not been utilized on a regular basis, there is a real lack of understanding of how they work, the complexity of them, and also what is required to deliver a study in a center with cell therapy, with cell and gene therapy. So we have, for example, done quite a lot of work thanks to our GMP team in bridging the knowledge that the hematologists already have in the space with the hepatologist so that they could learn what were the requirements that they had to have in their hospital, what were maybe just the additional hurdles that the cell therapy protocol had. For example, GMO committees or ATMP committees that some centers have. So that's definitely something that if we didn't discover it early enough, that would have definitely tripped us up. And we found there was like a real gap of understanding about how ATMPs work and what's needed for their clinical development. So those are the two things that immediately come to mind. And then I think we cannot fail to mention that the last couple of years, funding has been quite hard to come by. The overall environment has been challenging for funding. We've just closed a Series B a few weeks back, but that's been a real uphill battle to get to that point. So we're very proud we got there, but obviously we had to spend and invest a significant amount of time and effort into achieving that. So certainly, because cell therapy are expensive to develop, that is definitely a hurdle that unfortunately several companies in the last few years have not cleared.
Absolutely, I'm sure from the nods that everyone found that a relatable answer. Haley, I'm going to move over to you now with a question about R&D processes for cell and gene therapies, which obviously involve many specialized functions. What are the current methods and tools used in day-to-day record keeping and data collection in these R&D steps? And why do R&D teams engaged in developing cell and gene therapies need dedicated unified software solutions?
Yeah, so many of the teams that we work with are using various solutions such as Excel, paper notebooks, Google Docs, and then maybe different molecular biology tools. And then these tools are familiar to the teams and can work up to a certain scale, but we find that they become more and more challenging for teams to work with as they grow and capture more data. So with these disconnected systems, finding your data, sharing and linking that data across teams, and performing impactful analyses can all be challenging. So scientists need to design and track their sequences and constructs, and then utilize them in processes and capture the full experimental context. All of this data then needs to be found, shared across teams, transformed, processed, and finally analyzed. So this can create what can be labor-intensive and error-prone processes, especially when that data is stored in disparate systems and not standardized. So having all of this work from design to analysis on a single platform really streamlines the process and allows teams to derive more value from their data. So beyond being able to easily find their data, a unified platform can provide teams with benefits such as traceability, having that full experimental context of all of their samples, easily sharing that context across teams, and then accurately modeling the constructs they're actually working with all on a single platform.
And now, Tim, let's move over to you. What do you think is the biggest hurdle for cell therapy companies preparing for IND?
Largely, it's the CMC module, just getting all of the manufacturing sorted out and the QC. You know, it's challenging to make these drugs and to make them consistently. And so a lot of companies may not be prepared for the level of detail and characterization and data required to get an IND open. So yeah, that's the biggest hurdle in my mind. Clinical can have hurdles as well, but typically where companies struggle and where timelines get delayed is around the CMC.
Okay, great. Thanks so much, Tim. Neil, the next question I have is for you, which is, why has genetically unmodified T-cell therapy not seen wider adoption, even though it was first introduced decades ago?
So I think that the first real demonstration, you could argue a little bit about the impact of genetically unmodified T-cells, was from Don Thomas, who helped to demonstrate that when people were cured of leukemia with a bone marrow or stem cell transplant, that it was the immune system of the donor that really played a very significant role beyond the chemotherapy and the radiation. That was in 1979, so more than a few decades ago. The problem has really been, how do you develop that approach from a donor to a recipient and control the process so that you know exactly what you're giving, exactly how much you're giving, and can do it in a consistent and reliable way. Without getting too technical, one of the limitations of genetically unmodified T cells is that there needs to be a degree of matching between the donor and the recipient for something that we call HLA, which is what people get matched for when they have an organ transplant. It's very heterogeneous. We now have the tools where we can make a product where one and only one HLA is important in the T cell product. We now have tools where we can quantitate exactly how many of the cells recognize the targets, and I'd emphasize targets as opposed to one target, which gives us a redundancy and a breadth that you don't always have. And this now allows us to sort of bring this approach to people who either couldn't do it to a strong enough degree themselves, immunocompromised in some way, or couldn't do it fast enough themselves. And it's not really far-fetched to believe that we're not far away from being able to see a patient in the morning, get some blood work and things done, quickly. By the afternoon, actually treat the patient, watch them for, an appropriate period of time, and send them home to sleep in their own bed that night. So the tools of precision and quantitation and consistency are really now available to us. One of the nice things about T cells that I would add is that if a new variant of a virus, whether it's COVID or anything else, comes along, all you need to do is sequence that new strain, and with certainty exactly which of your targets are preserved and which of them are not. And I would say that in contrast to some other things, T cell targets are kind of stubborn and persistent, more so than some of the other targets really are.
Great. Thank you so much for that answer, Neil. So now, before we get to the end of this discussion, I wanted to offer everyone the opportunity, if you did want to add anything or comment on something that one of the other panelists had said, you're welcome to do so now.
So I think that one of the challenges that we all face, and it won't be something that we, perhaps deal successfully with the first generation of product, is in addition to safety and efficacy, to deal with affordability. Healthcare is growing increasingly expensive. The affordability and, again, that should improve over time is something that I will say really resonates very strongly with Ryan Sadi, our CEO. And it's important for all of us to keep our eye on this, and to work toward it. Some things in the beginning will certainly be very expensive. You can't help it. But we've got to dial it down so that it's affordable and can be used in a widespread way.
Absolutely. Thanks for that. I think the nods all suggest total agreement there.
I absolutely agree, Neil. However, there was a recent release from ARM, the Alliance for Regenerative Medicine, I think it was in August, where they did some really nice case studies where they tracked the cost of the product, which they are quite expensive, against the lifelong cost of treatment. And there's the significant not only cost savings, but resource savings as well for the health services. Yeah, absolutely. But certainly driving down cost of goods as well, Neil, to drive down the cost as well. And within Catapult, we're looking at automation and digitalization, because not only will that drive down cost of goods, it'll improve consistency and quality, hopefully. So I think that there should be a big push on that also, automating.
And if I can add instead something about what Haley was saying, I think actually digitalization across the board is going to be absolutely crucial for the development of the cell and gene space. We're currently going through this exercise in Resolution, and we did an internal case study on how many hours we could save if we had a digitalized integrated solution for data collection and analysis, and is mind-boggling. And it's really something that often go unnoticed. People think it's a lot of new skills to take on board, and therefore they become a little bit reluctant in the implementation phase. A lot of these projects do fail in the implementation phase. But I'm a firm believer that if we do not have an upskill in terms of IT skills and the digitalization of our businesses, it's going to be really, really challenges to grow at a reasonable pace and with reasonable costs in this field.
I just wanted to ask more, like, so we're talking about the digitalization and data, which makes a lot of sense. Where are there other places in the research and the commercialization, I would say, process that you see that can work more on saving costs, making things more streamlined and efficient?
I mean, Haley seems the expert, so I will let her answer, and then I can provide my perspective.
Yeah, I think we looking for solutions that really can grow with the team and fit a lot of different areas, and that's something we work with at Benchling as well. That we started really kind of niche in certain areas, and we started with just a few applications and functionalities, and then trying to think and grow to fit the full lifetime of projects from, again, that initial design, and now we're working towards bioprocess and more downstream efforts as well. So at least in terms of the solution like Benchling, we are trying to think, what are all the areas we can touch and help our different customers with? And also thinking around that digitalization of data, making our data AI machine learning ready too, is also really important. That if we have paper notebooks, that's going to be a lot more challenging and really limit kind of where we can go and the tools available, not only just in our space, but kind of coming out of all different areas of research.
Thank you. And now, Tim, let's move over to you. What do you think?
Yeah, it's important. The sooner that you can digitize your data and kind of set up your systems to collect and manage data electronically, you'll save a lot of effort, and both in the doing the work up front and then in the analysis downstream. So it's best to consider that and invest in that up front. Having an electronic quality management system is super important these days. It's nearly impossible to do it on paper in a way that has data integrity anymore. And so it's very important both on the EQMS side and then also for the manufacturing execution in terms of electronic batch records and or tracking and of chain of identity and chain of custody for materials kind of flying around the world. And it's particularly for autologous therapies where the product has to come from the patient and go back to the patient. You really need robust electronic systems and validate electronic systems to do that effectively.
So if I may just, add to that, I think that, teeing things up for machine learning, AI, automation is absolutely crucial. I know in terms of the work we do, there is a lot of background work that we do in the office with computers. There's a lot of manual things that, require a lot of personal intervention before we ever take, our potential targets to the lab for confirmation. And, one of the things that we're actively working on, as I'm sure all the rest of you are, is ways to automate that process, to try and streamline the process, learn a little bit about the human genome and how that influences T-cell targets, which it certainly does, so that we can move quicker and faster and in a much more generally expedited way. And I think that's obviously important to all of us.
Absolutely. Thank you, everyone, for weighing in on those different points. That's, I think, all we have time for today. I'm sure everybody could talk about this all day, but we're going to have to call it there. Thank you so much for your time today and for sharing your insights. I have no doubt that everyone will have found this very interesting at home. So thank you. It's been great to hear all of your insights today.
All right. Thank you.
Thank you.
Thank you.
Bye.
Thank you very much for that fascinating panel discussion. If you have a question for any of our speakers today, you still have a chance to ask by using the questions panel, and we'll be in touch after the showcase. Today's showcase is co-sponsored by Benchling, Sartorius, Applied Stem Cell, and Elegant Bio. We will now hear from Tim Largan, Chief Operating Officer at Applied Stem Cell, for a sponsor presentation. Over to you, Tim.
Hello, I'm Tim Largan, Chief Operating Officer for Applied Stem Cell. I'll be discussing CMC considerations for cell therapy developers as they prepare for an IND. Materials are critical and often the first thing the agencies will ask about. For cell therapies, donor screening and selection is critical. For allogeneic treatments, you need to ensure that donor screening and testing meets all the current requirements and guidance for your intended region. These tend to change over time as pandemics come and go or different diseases come and go. So make sure that you are using the most current ones for the region that you intend to be doing your studies or that you intend to expand to at the time that the donation is collected. For autologous treatments, donors used for demonstration stability batches should be as representative of intended patient population as feasible. Oftentimes, you're testing your drug in a diseased population, and for an autologous treatment, you may not be able to get donors with that disease state. So you should consider what that means in terms of your process, how that might impact the process, the differences, and try to account for it as much as possible. For cell banks intended for use in early clinical on through commercialization, you need to employ full GMP at the earliest stages of development. For reagents, raw materials, and components, use the qualified GMP grade materials whenever possible, not only reagents and key materials, but any material that touches the product fluid path. It's best to avoid human and animal-derived materials wherever possible. These have a lot-to-lot variability potentially, but for some therapies, if these are required, you should ensure robust documentation and consult the guidance and make sure that you have, certificates of origin or BSC, TSC statements. Sometimes inactivation methods are questioned by the agency, so make sure you're doing those the best you can and you have answers for those questions. For gene-edited products, you need to ensure the quality of your gene editing components are GMP from the beginning of development. On to process design and control. In general, process descriptions must be clearly defined with scientific rationale for all of the key parameters and quality attributes. Where safety and efficacy considerations allow, give yourself more flexibility in the early stages and tighten your parameters as clinical experience develops. In other words, don't lock yourself into a requirement on CO2 content in an incubation step that takes 3 hours, right? Probably doesn't matter to your product, so don't put it in your records that it's required. Give yourself room, because in cell therapy, the process is the product, and you really need to consider the commercial scale process and operations early in your development process. This means, like I said, about keeping parameters and specifications loose early on, because you don't know how difficult they will be to meet as you gain more experience with your process. And also process changes for scale up or scale out, or adding cryopreservation when you started with a fresh product. Those comparability studies can be very difficult. And as you get further into clinical development, the bar becomes much higher for statistical significance and for the agency's risk tolerance. So try as best you can. It's impossible to do it perfectly, but try to envision what your product looks like at commercial scale before you start your first clinical trial. On the QC analytical side, all release methods must be at least qualified at the phase 1. It used to be that the agency would give a little more wiggle room on this, but in recent years for cell therapy and gene therapy products, they've required that you explain in your IND the descriptions of the qualification activities you've done on each release method, as well as system suitability procedures that you have in place. Of course, safety methods must be fully validated by qualified labs before you get started. That's a must-have. Would also advise to leverage characterization assays early on to build process understanding. So not necessarily release or in-process control, but more process monitoring. And this is particularly important for potency assays. And a good plan is to start with a very large panel of assays that you think may give you insight into potency and mechanism of action. You can start with a large panel. I know it's time-consuming and expensive, but this way you can narrow it down as you learn which ones are important and meaningful and which ones aren't. And hopefully, whatever you have at phase 3 will tie nicely with efficacy, and you'll have a potency assay ready to go. So many companies have been caught not having this when they get to the stage of BLA, and it causes them significant delays and financial hardship. So in terms of agency communication, in your pre-IND meeting, you should be as prepared as possible before submitting the request. It is often difficult or impossible to get more than one pre-IND meeting. Initial questions from the agency are often boilerplate around donors and materials, so there's no need to panic if you've done those things correctly. Be very thoughtful about the content and phrasing of your questions and your briefing book ahead of the meeting. Identify the trickiest challenges and highest risks in your process and analytics, and anywhere where you need to deviate from something in the guidance or just because of the nature of your product or what have you, the nature of your process. But for those things where you must differ from guidance, propose a plan and a rationale, and then ask if the agency agrees. Once you've submitted this briefing book, you need to make sure that your key SMEs are available to respond, because this back and forth before the meeting is crucial for the company to get it right. So all hands on deck. And leverage your contacts with the agency. You'll have an administrative contact, like a project manager, that you can email with questions, but I would advise don't email them with every little question that you can find answers to in the guidance. Don't be a pest, but do leverage them. And then if you have other channels, use them judiciously. Reviewers from previous products, connections with the agency that you can informally ask their opinions on, that is acceptable. Just do it judiciously. And then submit your IND only after very thorough and careful review. Ensure you've addressed all questions and outstanding issues from meetings and correspondence, and then keep your ringers and notifications on and your SMEs at the ready, and expect last-minute communications and questions as the clock runs out. So with an IND, you submit, and then you have 30 days. The agency has 30 days to either ask you questions or put you on clinical hold. And so what usually happens is 2 or 3 days before that clock runs out, you get questions from the agency, and you have to respond to them quickly. And sometimes they'll respond to your responses. And so particularly in the last week of those 30 days, be at the ready and ready to call meetings and write up answers quickly. So if you've done these things, you have a good shot of getting your IND open and avoiding clinical hold, at least from the CMC side. So thank you very much for your attention, and I hope this was helpful, and thank you. Bye-bye.
Thanks for that presentation, Tim. Before we move on to the next presentation of the showcase, I'd like to remind everyone that this event will be available to access on demand on the DDW website, www.ddw-online.com. Next, we will hear from Victor Dillard, Vice President of Strategy and Operations at Resolution Therapeutics, who will talk about pioneering regenerative macrophage therapy in inflammatory and fibrotic diseases. Over to you, Victor.
Hello, everybody. My name is Victor, and I'm the VP for Strategy and Operations at Resolution Therapeutics. And today I'm going to tell you a little bit about Resolution Therapeutics and our work in pioneering regenerative macrophage therapy in inflammatory and fibrotic diseases, and how that fits in with some of the opportunities and barriers that I've observed in the cell and gene therapy landscape, and how we look at some near-term technologies to create efficiencies in developing cell therapies. Briefly, as a company, we're working on a platform called regenerative macrophage therapy, and I'll explain a little bit more what that means in a second. And we're tackling a very high unmet medical need, which is end-stage liver disease. With regenerative macrophage therapy in end-stage liver disease, we aim to transform patient outcomes in a patient population that is rapidly growing and that has no alternative treatment today. we're working specifically with engineered macrophages, and this engineering has allowed us to improve on the anti-inflammatory and anti-fibrotic properties of the platform, which we think is key to delivering these transformational outcomes for patients. And this is built on the back of 15 years of preclinical and clinical research coming out of the University of Edinburgh that has actually proven and demonstrated safety and efficacy of macrophages in patients with cirrhosis. So a very strong preclinical and clinical platform on which we're building a commercial product to take to patients with end-stage liver disease. But taking a step back, I wanted to just sort of do a simple mapping of the cell and gene therapy landscape and focus specifically on where I think the opportunities in cell therapy are today. We all know that cell therapies can deliver transformational outcomes. We've seen that with the CAR T therapies and the successes that those have had in patient populations who really had no alternative pharmacological options for their disease. And we've seen some fantastic data in the clinic of these CAR T therapies in some very aggressive forms of cancer. And actually what that's driven is a huge influx, of course, of companies developing cell therapies in oncology. So what I did is I looked at how many therapies were being developed across all development stages, including being marketed in oncology. And I grouped the ones that I think are sort of our first wave, our traditional cell therapy. So the CAR Ts, all of the NKs that came after that, the TCRs, the TILs, compared to what I call sort of the other cell types. So maybe your macrophages, your B-cell medicines, platelets, things like that. And what you can see is a real accumulation of over 2,000 trials and studies of what I would call traditional cell therapies in oncology versus just under 500 of these other cell types. So there's a real sort of question, which is, as a cell therapy company, if you're developing T cells in oncology, how would you differentiate yourself today? And I think that's a really, really big challenge. And therefore, for me, the opportunity lies beyond oncology and beyond the traditional cell types. Because already beyond oncology, we've started to see, following some recent very exciting papers about how CD19 CARs, for example, were effective in autoimmune diseases. A lot of companies have expanded their pipeline or even completely shift their focus into non-oncology applications such as autoimmune diseases. But again, staying on the same CAR T or T cell platforms. Where I think the real blue ocean of opportunity lies is where there are novel cell types in areas of very high unmet medical need beyond oncology. And of course, this is where Resolution plays today. Our first indication as a company is end-stage liver disease. It is a very large unmet medical need and represents a huge market opportunity to develop a product in. End-stage liver disease is the result of years and years of chronic inflammatory damage to the liver, either driven by etiologies such as MASH, alcohol, but also viral hepatitis. And it results in the liver losing its ability to perform its natural function. And this manifests in the form of decompensation events. These are clinical events that typically require admission to hospital, such as variceal bleed, hepatic encephalopathy, severe jaundice, or ascites. And patients with decompensated liver cirrhosis actually have a very poor survival. And you can see there on the top right that it tracks much like some aggressive forms of cancer, such as relapse refractory multiple myeloma or DLBCL. And the median mortality between the first decompensation event and the second decompensation event drastically drops from 2.5 years to just under 9 months. So there's a real need here to prevent patients from further worsening their disease, because after the first decompensation, having another one severely impacts survival, especially since patients today have no pharmacological treatment options. The only treatment option really is to wait for a liver transplant, and this waiting can often be fatal for patients. And this is actually a very big patient population. It's a very big one, and it's growing due to the growing incidence of MASH and actually the rise in alcohol intake. And when we make an assessment of also some of these earlier lines of treatment, tackling MASH, tackling some of the early fibrosis, even the GLP-1s, we see that it makes very little difference to the growth and overall size of the patient population over the next few years. So we've focused as a first indication at Resolution on an area of very high unmet medical need, but also a very large patient population. And what we're bringing to this patient population is regenerative macrophage therapy. Macrophages actually have a very innate ability to resolve organ damage, and this has been shown in liver cirrhosis specifically because they act on these two axes of the mechanism of action. The first is an anti-inflammatory mechanism of action, and that's really driven by the ability of macrophages to phagocytose and to eat and remove bacteria and necrotic tissue, which prevents the activation of downstream inflammatory cascades, but also their ability to recruit blood circulating monocytes and polarize them to anti-inflammatory macrophages, thereby creating a perpetual anti-inflammatory effect that is needed to allow for the liver to regenerate. At the same time, patients with liver cirrhosis have a very high amount of scarring or fibrosis, and the macrophages actually have a mechanism of action that allows them to tackle that by breaking down the extracellular matrix and remove that fibrotic scar tissue, but also suppress the formation of new scars and suppress fibrogenesis by deactivating and downregulating hepatic stellate cells. And this is key because it allows hepatocytes to regenerate, and this is, of course, one of the key drivers of liver regeneration, which over time allows the liver to rebuild the hepatic reserve it needs to perform its function. So macrophages have this innate ability that we're leveraging here to help drive regeneration in the liver. And I mentioned the sort of work that Resolution is built on. We believe that we have a very strong route to clinic because macrophages have actually been shown by Professor Stuart Forbes at the University of Edinburgh to have a strong beneficial effect on survival in patients with advanced cirrhosis. And so on this slide, I'm just showing the results of the MATCH study. This is the 2.5-year results that were presented earlier this year at the EASL Congress in Milan by Professor Forbes, where non-engineered regenerative macrophages had a significant impact on survival and transplant-free survival. And so this gives us confidence that the platform that we have is really fit for this high unmet need. But we're taking it further, and at Resolution, we're engineering macrophages, and this is our core program. This is RTX-001. Like the product used in MATCH, it's an autologous monocyte-derived pro-regenerative macrophage, but it has been engineered to have a more potent and durable clinical effect. And so some of the data that I'm showing here is the comparison of the non-engineered macrophages that delivered the clinical benefit shown in the previous slide versus the engineered macrophages, our lead asset, across the mechanism of action. So improved fibrogenesis suppression, maintain very high phagocytosis, improve the recruitment of monocytes as well as the polarization of those. And what we saw in vivo also was that these macrophages actually regressed fibrosis. So this is in mice models of liver cirrhosis. So for us, this is really the combination and the intersection of a novel cell type with a clear mechanism of action, with a strong preclinical and clinical research base, going into a very high unmet medical need and patient population outside of oncology. So I sort of use this as an example of where I think the opportunity is in cell and gene therapy. And one of the ones that, of course, we're very excited about is macrophages in end-stage liver disease. But beyond that, inflammatory and fibrotic diseases, and I'll touch on that in a second. The other thing I wanted to also just touch on is, of course, now we've identified this big opportunity, we've identified this novel cell type, but there are barriers in developing treatments in cell and gene therapy. And the three that I just wanted to flag were manufacturing, equipment, and development. The three barriers that I wanted to just touch on briefly in the development of cell and gene therapy products were manufacturing, equipment, and development. Specifically, manufacturing and the tension that companies face, which is how much to invest in improving our manufacturing process, in developing our analytical strategy, and how early to do this, whilst carrying, of course, the product and the clinical risk throughout. And this is a real dilemma for all companies and makes developing cell and gene therapies very challenging. At the same time, a lot of these products are spun out of academia, and academia will have, of course, developed a manufacturing process, but the minimal required to get the product tested, for example, in a clinical trial, often not suitable for a commercial process, a pivotal trial, or definitely a commercial launch. And so at what point do we start to invest in upgrading and improving that manufacturing process? At Resolution, we've done this very early. We were very heavily capitalized and well capitalized by Syncona to invest in that early. And as part of our recent Series B round, a large fraction of the funds raised also will be going to improving our vein-to-vein time and reducing our cost of goods, because we recognize how critical this is in tackling a large patient population and making this product a commercial success. So we've taken an approach to invest early and invest heavily in improving manufacturing robustness, in reducing cost of goods and vein-to-vein time of the product. The second piece I want to touch on is equipment. What we've often seen in developing manufacturing processes for cell therapy is fantastic innovation from equipment providers, but often in a sort of quote-unquote black box mode, where the equipment is provided, but any interaction with the raw data, any tweaking of the parameters is very much controlled by the original equipment provider. And I want to flag this as a real limitation and a barrier to accelerating the development. If you take an analog in the software world, people would publish code online on GitHub and on platforms and actually allow others to build rapidly and iterate and improve on software code. All of the large language models, they've published how they develop these, they've published the source code, and this allows for a pace of innovation that is orders of magnitude higher than what we're seeing in cell and gene therapy. And this is critical because we are then tied to specific equipment providers. We are also tied to specific file formats, connecting with other lab instruments, ELNs, data aggregation infrastructures. All of those are then really limited. And in a world where everything is being digitized, I see this as a massive holdback. And so I think as an industry, we can do a lot better to develop equipment that allows companies to take what has been maybe developed, for example, for T-cell therapy, and innovate rapidly with the provider to make this suitable for macrophages, for B cells, for platelets, you name it. The third and final piece, of course, is the cost of development. These are very expensive drugs to develop because, of course, manufacturing batches for patients in early phases is also very expensive. But the whole logistics around delivering a cell therapy product to patients in tertiary centers can be very costly and very complex. And if on top of that you need to carry out perfectly double-blinded randomized controlled trial, or even sometimes maybe placebo-controlled trial, you're adding to a really very high cost of development. So I just want to finish off with this last slide, which is around where I think the near-term opportunities to create efficiencies in cell therapy are. And there are plenty, and I just wanted to flag two of them, maybe for thought, as a thought-provoking exercise, because I think these are very near-term and actionable. The first for me is that the opportunity to create an end-to-end digital infrastructure is there from day one. There's no need to wait until the company has reached a certain, quote, level of maturity before implementing that. I think, platforms out there that have innovated over the past 5-10 years are ready to start accompanying cell therapy companies from day one to create an end-to-end digital infrastructure that allows us to collect the data right from the start, right from the very first leukopack that you receive into the company and that you process through. There's no reason why you shouldn't start capturing all the data in a format that allows you to make an end-to-end comparison. Because, of course, here we're dealing with the patient, which is the process, which is the product. And so unlike a small molecule, as a cell therapy company, it's really critical for us to integrate all of the data that we're generating around our process and our engineering in research, as well as what's happening in manufacturing, whether GMP manufacturing or the process and analytical development, with the clinical. That means that you need an integrated IT strategy, and you no longer can just have sort of R&D IT and then clinical IT and then corporate IT. You need a holistic an integrated IT strategy that really leverages absolutely everything from early research through to the clinical data. And I think this is achievable today, and I think there are companies, that are selling great platforms, services, products that can enable you to do that. The second thing that I want to put out there is I think there's a real opportunity to build robust external control arms from real-world data. Robust because the quality of the data and how we're capturing it with this transition into digital health records is really increasing. The FDA has actually recognized the value and the use of external and synthetic control arms. They've put guidelines, they've put guardrails around when to use it, and they've also, still maintained that the randomized, blinded, placebo control approach is still the best. However, we have to recognize that for cell and gene therapies, this may just not be the case. So how do we build the right statistical frameworks and analyses so we can leverage external control arms and still ask a very pure and very high quality statistical question without cherry picking data, without sort of creating the external control arm that perfectly matches this is not good clinical science. What we want to do is we want to mimic what can be done in an actual, randomized control trial, but leveraging what's actually being collected out there in a real world setting. This could potentially cut development time and cost by 50% and also improve probability of success of trials because we come into them with a much deeper disease understanding up front and also established relationships with key opinion leaders and sites. So I'll end there. These are for me the sort of near-term opportunities that I think are within reach. They require industry collaboration between developers, between equipment providers, between the software world, but also with hospitals and with regulatory authorities. All of this data sits in hospitals. It sits behind firewalls. It's of course being protected for the benefit of personal and patient privacy, which is critical. But we have to leverage it in a way that allows us to derive value that we can bring back to the healthcare system. There's an opportunity for hospitals to monetize this. There's an opportunity for patients to get better medicines more quickly. There's an opportunity for regulators to get a better understanding of diseases. I think the opportunities are ripe for the taking. So with that, I'll finish off. I hope you found this presentation interesting and the blend of how at Resolution we're pioneering regenerative macrophage therapy in inflammatory and fibrotic diseases, but also how we look at some of the opportunities and barriers in cell and gene therapy. I'll be happy to take any questions now. Thank you.
Thanks for that, Victor. We'll now move on to some audience questions. The first one we have for you is, Resolution is developing a cell therapy treatment for a very large patient population. What are the considerations when going after such a big unmet medical need from a cell therapy perspective?
Yeah, so at Resolution, one of the things that we think plays in our favor is that as a therapy, what we've seen from the match data is the macrophages are very well tolerated. And so we see this now as outpatient compatible. So macrophage cell therapy has the potential to be administered in secondary sites. Additionally, there is no requirement for any bridging therapy, any preconditioning. And so I think for me, this is a real key aspect to ensuring that as a therapy, you're maximizing your potential for broad adoption. Because if you're limiting yourself to referrals to tertiary centers and centers of excellence, inpatient settings, long infusion times with complex bridging therapies or preconditioning, of course, the uptake is going to be much more limited. There's going to be a funnel of patients coming into that stage of therapy. And I think that's a real challenge for very broad patient populations. Whereas if you can develop a therapy that can be administered in an outpatient setting, and that doesn't require all of this additional work, then you've taken away this funnel, and actually it's, as a company, you can then focus on your ability to manufacture and deliver batches in time into the volume that's required to meet the demand. At the same time, I think it's important to recognize that, in the case of end-stage liver disease, there is very limited competition, if you will, for patients at that final stage. There are no pharmacological treatments. all the earlier lines of treatment have very limited antifibrotic activity. So actually a lot of patients still will progress with their fibrosis and end up in end-stage liver disease. And there's a funnel there where patients have nowhere to go. So there's sort of an accumulation of patients at that final stage. In some of the advanced, oncology applications, there have been some terrific innovations in bispecifics, for example, in radioligands that actually are competing quite heavily with some of the cell therapy. So I think it's important to consider when going after such a large patient population, is there a pocket, is there an ability for us to go after a patient population where actually there is, really no alternative, and there is, from a therapy perspective, we can ensure a broad adoption.
Great. And the last question we have for you is, when developing a cell and gene therapy product, should the focus be on a single asset in a single indication, or on demonstrating the broader platform potential of the technology?
My view and what we've done at Resolution is get very product focused from the start. So we were very much focused on developing RTX-001 and getting that to be clinical ready, by our Series B cycle. Now that we've raised Series B, now that we've launched RTX-001 in its phase 1/2 study, now that we've started enrolling patients, we can start to look at expanding the platform. And I think you're always sort of playing, this tension of losing focus on your core product, because it is hard and it takes a lot of work, and it takes really one team effort to get these products to the clinic. And so if you're diluting that across multiple indications, then you're reducing your chances of success. But at the same time, the further along you go in the development journey, the more risk is carried by that one single asset. And so you want to go and de-risk this. You want to go and test, look, can this go and be applied to other indications? If this is truly transformational, we almost have a duty to go and test, like, could this help other patients in other inflammatory and fibrotic diseases? And so at Resolution, the way we've tackled this is to have a really strong focus throughout our seed and Series A period on getting RTX-001 to the clinic. Now that we have done this, we've opened up, some funds as part of our Series B, but it's really not the lion's share. The lion's share really remains on RTX-001, but some funds to go and explore regenerative macrophage therapy and other inflammatory and fibrotic indications. So the answer to the question is, a blend of both, and I think it depends on timing, and it depends on funding situation as well.
Thanks for that, Victor. Now we will hear from Hayley Merritt, Scientific Solutions Consultant at Benchling, for a sponsor presentation.
Hi everyone, I'm Hayley Merritt from Benchling, and today we'll be discussing how Benchling can support cell and gene therapy groups. First, I want to cover a common challenge we see amongst many cell and gene therapy teams. Often, teams will be utilizing a variety of disparate point solutions to manage their data. So this could range from Excel to Google Docs to paper notebooks and molecular biology tools. And while these solutions may all be powerful on their own, they're not connected or standardized, which can lead to additional challenges for teams. Finding data, especially if I was not the person to capture that data, and linking that data back to the relevant samples and experimental context can become increasingly difficult as organizations scale and remain on these disconnected systems. This will lead to delays in work, incomplete data sets, and gaps in decision-making. By utilizing a unified platform like Benchling that can capture a team's work from end to end, teams can streamline these tedious tasks and get more out of their data. From in silico design to final analysis of their data, a centralized platform will capture all of this work and allow teams to focus on their science and derive more value from their data. Today, we'll be reviewing some of the ways Benchling addresses these challenges and can fit the unique needs of cell and gene therapy groups. Let's dive in and take a look at some of the end users of our platform. First, we'll focus on the challenges faced by R&D scientists in the cell and gene therapy space. With a disconnected system, a research scientist on our team may be faced with challenges around entering, finding, and utilizing their data. With systems that are not scientifically aware, capturing sequence-specific data isn't possible, and finding those samples is challenging. Capturing results in a standardized format and associating those results with the appropriate samples can also be difficult as teams track their data across multiple systems. And finally, our scientists often find themselves using clunky and non-intuitive tools, taking extended time out of their day to enter and search for their data instead of focusing on their science. Let's go into the platform and see how Benchling can address some of these common challenges. As a scientist working in cell or gene therapy, sequences and plasmids may be critical to my work. Here, I'm looking at a plasmid that we are tracking in Benchling. All in one platform, I can capture and find both experimental and biological information about this plasmid. I can manage all of my sequences and designs with a full set of molecular biology tools, and in the same platform, start to build out relevant lineages with my other samples. Let's check out the CAR associated with this plasmid. Benchling is a platform that is built to model cell and gene therapy, and we have experience and best practices to help teams track the information and parts that are important to them, such as their domains, spacers, or linkers. And we can track all of that important information in the same platform where we can easily capture experimental context. Traceability is critical for our teams, and in Benchling, I can quickly find the full experimental history of my samples all in one place. Whether that's the experiments this sample has been used in, results captured against this sample, or available inventory and batches of my samples. Now let's go ahead and take a look at how our research scientists can actually capture all of this data in the platform. Here's a notebook entry that I previously was working on. The ELN feels very familiar to teams, like other word processing software, so it would be easy to start using in their day-to-day. These entries go beyond simple paper on glass, because what we capture here is connected across the platform. By capturing our data and experimental context in the same platform as our molecular biology tools and sample management, we never have to worry about copying and pasting our data across our different systems or using error-prone methods to connect data back to relevant samples. We have some user-friendly features like mentioning our team members for easy collaboration, dragging and dropping in files so we can easily view and reference different types of files or images on the platform, and then specific tables and tools that I can search for samples or data across the platform without ever leaving my notebook entry. And the tables within these notebook entries help streamline and standardize experimental data capture for our scientists as they work through their processes. Right from my experimental entry, we can see here how I entered samples into the platform, moved those samples to my inventory. So here we can see a box filled with some tubes, and then captured results against those samples, all without leaving my notebook entry. And then, as we saw just a few minutes ago, all of the data captured in our notebook entries is traced across the platform. So clicking back into one of these samples, we can see the results that were captured in that notebook entry against the sample. And we can also see for the batch of those cells, all of the available inventory we created. So those different tubes. So all of these features come together and make it really seamless for our scientists to go back and find what they need and focus on their science. Next, let's change hats and dive into some of the challenges our R&D team leaders might face without a centralized platform. Our team leaders are concerned with taking all of the high-quality data our scientists generate and making impactful decisions based on that data. But with disconnected systems, this can be difficult. Our team leads want to more easily track critical milestones and metrics to improve their decision-making processes and improve operational efficiency. They also want to ensure that the data generated by the team is analysis ready, so it can be utilized to keep projects moving forward. With outdated systems, connecting to analytical AI and machine learning pipelines becomes increasingly difficult. I have previously gone into the platform and entered some data, and now we're seeing what we can do with that data when we capture it in a unified platform. Benchling makes it easy for us to visualize our data, answer the questions we want answered, and then take action on that. As we just saw, all of the data captured is tracked across the platform, allowing us to generate these dashboards to make quicker and more informed decisions. So these dashboards can not only capture experimental insights, as we see here, but also operational information. So team leads can identify gaps or delays in their processes and work to improve efficiency across the team. Finally, let's see how we can generate analysis-ready datasets right in the platform. Our scientists have diligently captured their data in the platform, so our team leads want to ensure we're using that data to generate insights. Data can be entered, as we previously saw, with results tables, or through direct integrations with instruments our scientists are using in lab through Benchling Connect. Here, I've queried data from across the platform and my samples to compile a data set. This data is now analysis ready, so I can seamlessly connect it to our analytical pipelines. So whether that is through tools that are external, such as JUMP, so here we can see I can connect to external tools, or utilizing some of our analysis tools directly in the platform, I'm seamlessly able to find and utilize all of the relevant data and apply it and analyze it. So here, just to see, we have a cell viability assay that we're visualizing, and then just another example, we can see some ELISA data that we use to generate a linear regression and a heat map. So having our data structured and standardized on a single platform allows our teams to also be AI and machine learning ready. As new tools and advancements in technology continue to emerge, we want to ensure our teams are able to use them to their full advantage and continue to advance at the same rapid rate. So I hope we showed today how Benchling can help our cell and gene therapy teams. So with our scientists, capture data and save time, and then our team leaders to go in and make better decisions based on that data.
Thanks, Haley. Up next, we have Dr. Neil Flomenberg, Chief Scientific Officer and Global R&D Lead at Tavogen, who will talk about targeting viral infections in cancer with cytotoxic T lymphocytes. Over to you, Neil.
Hi, I'm Neil Flomenberg. I'm the Chief Science Officer and Head of R&D for Tavogen Bio Holdings. The first demonstration of the effectiveness of immune cells in preventing infectious disease was Edward Jenner's use of cowpox material to successfully prevent smallpox in humans. In the cancer space, one of the best demonstrations was the graft versus tumor effect demonstrated by Don Thomas and his associates. They showed that the immune effects of the donor marrow were extremely important in curing patients of their underlying blood cancers above and beyond the impact of the radiation and chemotherapy that were used prior to the transplant. With regard to market size, vaccinations are broadly used across the world. In 2022, there were 12.7 billion doses administered. In the same year, the number of T-cell infusions was far smaller, 480. This has expanded significantly in the last several years, with the numbers now reaching into the thousands. But this is still a far cry smaller than the billions of doses of vaccine that are used each year. The best known T-cell therapy currently on the market is the CAR T-cell. This is a genetically altered cell where a new receptor is introduced into the cell. This requires collection of blood, typically from the patient into whom it's going to be administered, the introduction of the new receptor, their cell expansion, and subsequent infusion back to the patients approximately 3 weeks later. The artificial receptor which is introduced is in many cases on both malignant cells and their healthy counterparts, and both can be eliminated by the CAR T. This introduces the requirement that the healthy cells can be done without for a period of time safely and still be tolerable. This is true for some targets, but not necessarily for all. In contrast, the genetically unmodified cells that we refer to here as Mother Nature's CTLs have a major function of killing infected cells. They can also play a role in malignancy as well. When a cell is infected, new proteins are produced. These are degraded and expressed on the cell surface where they can be seen and recognized by the cytotoxic or killer T cells, which then, through a variety of mechanisms, produce cell death. The importance of this is that each viral infected cell becomes a virus production plant, and it's essential to control the infection that these cells are eliminated and replaced with normal, healthy cells by the body. One of the differences here is that there is recognition of the sick, either infected or malignant cells, but not their healthy counterparts. The breakdown products, the peptides which are recognized, are presented by a molecule known as HLA, which are the determinants that we actually match for in the course of a transplant. Because of the fact that people vary in terms of their HLA content, some degree of matching between donor and recipient is required, and a panel of cells rather than a single target is necessary for this product. So in contrasting these two approaches, in the unmodified T cells, we need, again, to make a panel, which we're hoping artificial intelligence will help to accelerate. We need to be able to target all the viral substrains, and this is a bigger problem for older viruses which may have coexisted with human beings for thousands to millions of years. We've tried to illustrate this in the figure. You can see in the left, the CTL grabs the target on the surface very nicely. You can see that in the cell on the right, the target is now a different color, but also a different shape and fits in the receptor a little less well. There are certainly fewer targets in cancers than in viruses. This is something that there are, tricks and tools that we can hopefully get around. There is a need to avoid malignant cells turning off this process by inducing an artificial T cell exhaustion. And the other issue is that allogeneic cells, meaning cells from a third-party donor, do not last forever in the body. When one comes to the genetically modified CAR T cells, there are other side effects which are not typically seen with genetically unmodified CTLs, including cytokine release syndrome and neurotoxicity, which need to be dealt with and require at least proximity to a tertiary care center. We increasingly will need to find targets which do not affect normal cells as well as the malignant counterparts. One of the areas of work is to try and produce off-the-shelf products, meaning to use allogeneic cells rather than the patient's own cells for each product. Again, we need to avoid turnoff by malignant cells, and we have the question that allogeneic cells do not last forever in the body. This illustrates the complexity of the viruses that are out there. While not each of these is a unique isolate, this illustrates the number of viral sequences which we have downloaded from various sources across the internet. You can see that there are nearly 4 million COVID sequences, ranging down to several thousand RSV sequences. And this is, again, where we're hopeful that computer support and AI techniques can get the job of screening done faster than people. We have successfully performed a phase 1 study of the CTLs that we've generated for COVID-19. It was extremely successful in terms of safety. We did not really have any significant adverse events in the course of the study related to the cells. There were great efficacy signals as well. There was no evidence that the patient's own immune response was in any way suppressed by giving the cells. And we found evidence that the cells persisted for more than 6 months after administration. We are focused on giving these cells to high-risk patients who are either unable to make a strong enough immune response to get rid of the virus or unable to do so in a sufficiently rapid time frame in order to be successful. Now, beyond helping these particular individuals, there is suggestion that treating this group may help to avoid disease resistance and mutation, which may affect both vulnerable and less patient vulnerable groups down the line. On the right side of the slide, we've shown an example of a reference. There are others that suggest that new variants disproportionately emerge from such immunocompromised individuals who have a longer period of interaction between the virus and their weakened immune system, thus providing the window of opportunity for the new variants to emerge. What we're hoping to evolve as the therapy becomes more widely used is to continue to demonstrate the safety of the infusion. Again, develop further data with regard to the effectiveness and durability. And I should indicate that although the cells are not the same as we're talking about, restricted by a single HLA, there are more than 500 patients reported in the medical literature that have gotten this sort of therapy with a great track record of safety and effectiveness. Obviously, one of the things that we want to do is keep costs down so that this is an affordable treatment. We are hoping to make it far easier for patients and their families. We're hoping that with regard to something like COVID, a patient could be seen in the morning, HLA and other necessary information, blood could be drawn, results back by the afternoon, the patient treated that afternoon, observed for a brief but appropriate period of time, and then home and sleeping in their own bed that night. If we can pull all of these things together, which we're comfortable that we can, this should provide more widespread access to this sort of therapy.
Thanks for that presentation, Neil. We'll now move on to some audience questions. The first one we have for you is, why has genetically unmodified T-cell therapy not seen wider adoption even though they were first introduced decades ago?
A number of scientific and manufacturing developments have helped to make T-cell therapy far more analogous to conventional drug therapy. We're now able to make products that are restricted by one HLA only. We're able to know exactly which peptide targets are being recognized by this particular product rather than having a less well-defined set of targets that are produced in a peptide library or some other similar circumstance where you don't know exactly where the targets are. We have techniques so that we can quantitate the contribution of each of these targets to the overall product, and we can produce a product that's at least 70% active T cells in some circumstances, with ideas that we believe will help us to get into the 90s. These issues, we think, will move this sort of therapy from something that's a little bit hard to quantitate and be sure about to something that's very precise, the same way that one would understand a dose in other kinds of circumstances. And this will allow its more effective utilization compared to other approaches.
Thanks, Neil. And the second question for you is, how can the costs of T-cell therapy be moderated to encourage its broader application?
The costs of T-cell therapy can be moderated in at least two major ways. Number one, we produce hundreds of doses in our current iteration from every cell donor. We would hope that as we continue to gain experience, this number will continue to grow, and the more doses that are produced per batch, obviously the less expensive, that the product would be. The second issue is what I alluded to in terms of being able to treat a patient in a doctor's office or some other low acuity setting rather than needing to be tied to a tertiary care center, worrying about the need to admit the patient and other sorts of things that have a dramatic effect on increasing the cost of the treatment. Between the two of these, we believe that genetically unmodified T-cell therapy can remain extremely low and foster the broader application of this modality.
Thanks for that, Neil. Up next, we'll hear from Galit Meshulam-Simon, Associate Director of Commercial Applications at Elegant Bio, for a sponsor presentation.
Hello and welcome to the showcase. My name is Galit Meshulam-Simon, and I'm Associate Director of Commercial Applications at Elegant. Thanks for joining us to learn more about Elegant next-generation gene synthesis technology and how it enables the rapid production of high-fidelity DNA in an entirely cell-free process. Elegant and Finia DNA support advances in manufacturing and delivery system for cell and gene therapies at scale, addressing key bottlenecks at the supply chain. Elegant was founded in 2017 by Matt Hill to address the growing need of scalable, versatile gene synthesis. Elegant technology overcomes the limitation of conventional gene synthesis platforms that rely on cumbersome cell-based methods, where short gene fragments are assembled and cloned to correct synthesis errors and to obtain yield. Early in 2023, Elegant launched Finia DNA, providing researchers with double-stranded linear DNA up to 7 kb. Our DNA is NGS verified and shipped as fast as 6 to 8 business days. Conventional gene synthesis relies on a hierarchical build process. It starts with oligosynthesis from nucleotides and then stitching or assemblies of these low-quality, short, typically smaller than 2 kb DNA fragments into subunits. The subunits are being integrated into the final construct via methods such as Gibson or Golden Gate assembly. The assembly is a long, repetitive, and error-prone process. Once assembled, the final population of molecules contains a portion of sequence error that are incurred during the build process or even originated from the initial building blocks. In order to overcome that, the entire population of assembled DNA molecules is being cloned into plasmid and transformed typically into bacteria, and colonies are selected and sequenced just to isolate the sequence-perfect gene. Typically, larger scale cultures and plasmid purification steps will be needed to enable the required yield. This is a lengthy process, it's time-consuming, costly, and error-prone, and can take several weeks and sometimes even months to get to the big construct which is needed. Furthermore, cloning in cells has additional limitations, such as potential gene toxicity and contaminations from leftover cells. Based in San Francisco Bay Area, Elegant developed a patented cell-free cloning technology to overcome the limitations of traditional cell-based methods. We synthesize near clonal quality double-stranded linear DNA that is longer and with higher complexity than offered by conventional gene synthesis provider. It can ship within a week. Our production platform is highly scalable to meet the DNA demands in healthcare application, including cell and gene therapy and mRNA vaccines, without significant investment in resources and infrastructure. The speed, length, and complexity of Finia DNA has set the bar for next-generation gene synthesis. Instead of paying more and waiting weeks for long and complex DNA to be cloned in a plasmid, Finia DNA gives you up to 7 kb high-complexity DNA with NGS-verified quality, and all that without the wait. On the right-hand side of the slide, you can see our extremely low error rate, which is 1 to 70,000 per base pair. This is an average estimate accuracy of 99.999%. which is much lower in comparison to other linear DNA providers. In general, with our QC spec, we expect over 80% of gene synthesized to be full length. And out of that population, 95% should be sequence perfect. At this level of quality, it means that it's typically enough to pick 1 or 2 colonies to get the sequence perfect gene. And this is, of course, something that saves you a lot of time and cost. Elegant technology has been applied in application across various fields, including synthetic biology, protein engineering, cell and gene therapy, and agriculture. There are many applications across cell and gene therapy, so whether you are developing stable cell line like viral vector or for CRISPR technology, or whether your research is more about transient transfection development, like mRNA vaccines, antigens, and more, Elegant's DNA can be used quickly to provide you long template DNA with high quality. In the last year and a half, we've been growing rapidly with loyal customers in biotech, pharma, and ag bio, and we have only just started. Earlier this year, Elegant signed a multi-year collaboration with GSK that enables them to leverage Elegant's cell-free DNA production technology to develop DNA-based medicines and vaccines. Infinite DNA technology accelerates the build cycle. In this example, for AAV vector build, the conventional approach could include order of variable and constant regions, potentially from different vendors, internal assembly, cloning, picking, and sequencing to reach the final construct. This process is lengthy and could take 3 to 4 weeks and sometimes even more. By using Infinite DNA, the full insert can be ready within 1 to 2 weeks, completely cell-free. At this point, it could be used as linear fragment or could be used to assemble and clone and get your plasmid construct all within about 2 weeks. The time and cost saving are significant, and so is the reduction in immunotoxicity risk from cell traces. Another application is mRNA vaccines, which has shown their value for a broad range of therapeutic applications ranging from cancer immunotherapy to infectious diseases. Additionally, efforts are now being devoted to use IVT mRNA technology to other uses in cell therapy, such as CAR-T, TCR, and gene editing. Typical mRNA development cycle will start with assembly of DNA fragments, cloning, sequencing to find the perfect sequence gene, and will continue via master cell bank production and larger scaled plasmid production and purification to obtain the required yield for IVT reaction. Then the plasmid will typically be linearized and ready for the IVT reaction. Using Elegant and Finia DNA, you can get the linear double-stranded high-quality template very fast, add the poly-A tail, and be ready for in vitro transcription reaction. By simplifying and streamlining the mRNA synthesis process and bypassing the traditional cell culture, Elegant is enhancing process efficiency, lowering the risk of cellular contaminations, and overall reducing complexity and enabling faster turnaround time to refine candidates based on test results. We are continuously innovating to accelerate genetic medicines, and we have new products that are coming this year and early next year to further streamline and accelerate your workflows. Infinia DNA is available now directly by ordering from our portal at elegantbio.com with extended scope of complexity, including ITRs, LTRs, promoters, long homopolymers, short and long repeat regions, regions with high and low GC content. Later this year, we plan to launch Infinia Plasmid DNA with Infinia DNA cloned in standard backbones. This product will enable long, complex plasmid with shorter turnaround time. In early 2025, we plan to launch IVT-ready DNA. This is a linear double-stranded DNA template where the poly-A tail will be there already, and it could be at length as high as 160 base pairs. In the next 2 slides, I would like to show some case studies with our early access partner and client. The first one is around viral vectors production. collaboration with Asimov, which is a company creating a synthetic biology platform that integrates cells and software to enhance the design and manufacturing of advanced therapeutics. Their software facilitates the design, simulation, and optimization of genetic systems for viral vectors and biologics and mRNA therapeutics. And their aim is to improve the manufacturing outcomes. Collaborating with Elogen, Asimov efficiently synthesized complex DNA sequences essential for constructing plasmids used in the production of advanced therapies. The second case study is about the use of Elogen DNA for mRNA production. While the importance and promise of mRNA-based approaches are growing in different fields of personal medicine, vaccines and cancer immunotherapy, there are still many open questions about the effect of mRNA impurities that can occur during the IVT reactions on the functionality and stability of the produced mRNA. After establishing our process for IVT-ready DNA templates, Elogen had few initial validation experiments with early access clients and confirmed uniformity of the mRNA with different poly-A tails. With that, we proceeded to test the quality and functionality of those templates with early access partner. In a paper recently published in the Journal of Analytical Chemistry, Genentech Cell Therapy Engineering and Development team is using Elogen linear DNA templates synthesized with 3 different lengths of poly-A tails, from 90 to 160 nucleotide, and they've used that as a starting material for their IVT reactions for EGFP production. Their goal was to assess the impact of mRNA impurities on the production and stability of the EGFP in T cells. After analysis of the construct and one purified mRNA construct, and showing that there is a similar full transcript of mRNA, which you can see in the left figure, they used the construct to evaluate the expression and stability in T cells. The results, as shown in the right-hand side graph, demonstrate correlation between the poly-A length to the expression level of EGFP in cells. In conclusion, we presented today the capabilities of Elogen next-gen DNA synthesis technology to advance many applications in the cell and gene therapy fields. Our aim is to empower our partners and work together towards their goal. So please contact us with any questions or requests. Thank you.
Thanks for that, Galit. Lastly, we'll hear from Hina Kampwala, Principal Scientist of Product Development at Sartorius, for our last sponsor presentation.
Hello and welcome to this webinar presented by Sartorius. I'm Hina Kampwala, your speaker, and today I am excited to share with you some innovative transfection solutions that are driving advancements in cell and gene therapy. But first, let me introduce myself. I'm a principal scientist in product development based at our Bioanalytical Center of Excellence in Ann Arbor, Michigan. I'm an immunopharmacologist by training with a decade in drug discovery, and it was really this experience that provided a catalyst for a PhD before joining Sartorius in 2015. And for almost a decade now, I have been specializing in the development of innovative reagents and applications to support our bioanalytical instruments. Now, moving on to a little about Sartorius. We are a global leader in the life science industry with over 30 years of expertise in cell and gene therapies. With 60 plus locations worldwide and around 14,000 employees, our mission is to drive technological breakthroughs that empower scientists and engineers with the tools and processes for the development of better and more affordable therapies. Our solutions have supported over 200 drug candidates and are integral to the manufacturing of over 15 commercialized cell and gene therapies. And it is really our commitment to innovation that has allowed us to drive advancements in this therapeutic area. And with that, I'd like to introduce you to PolyPlus, now part of Sartorius. With over 20 years of pioneering innovation in cell and gene therapy and an ever-expanding global presence, PolyPlus Sartorius are at the forefront of industry. The solutions span the entire breadth of the cell and gene therapy workflow, all the way from life science research transfection solutions to the manufacture of GMP grade proteins and antibodies for therapies, making Polyplus Sartorius your perfect partner for all your cell and gene therapy needs. And so for the rest of this talk, I'd like to showcase our cutting-edge life science research solutions and hopefully demonstrate to you how these can help accelerate your cell and gene therapy research. But first, let's begin with an overview of the portfolio. At a glance, we offer a diverse range of transfection reagents that are tailored to meet your specific application needs. We cater for a range of molecules as well as a wide array of target cell types, all the way from cancer cells to hard to transfect cell types such as stem cells. The entire portfolio has been designed for ease of use, high efficiency, reproducibility, excellent cell viability, and importantly, cost effectiveness, all without compromising on performance. Before I take a deeper dive into some of these reagents, I'd like to briefly mention that at the end of this webinar, I can show you how you can request free samples for many of these reagents. So the first reagent that I'd like to highlight from this portfolio is Jet Optimus, a gold standard DNA transfection reagent that is highly efficient in both easy and hard to transfect cells. It comes with an optimized ready-to-use protocol that ensures excellent cell viability and morphology. And as shown by the graph and the images on the slide, it is suitable for a wide range of cell types, significantly outperforming leading competitors for both transfection efficiency and for its gentle effect on cells. Now, to avoid genomic integration of DNA, you may wish to transfect with mRNA instead, and that's where our Jet Messenger mRNA transfection reagent becomes extremely valuable. As one of my personal favorites in my work on transfecting T cells, Jet Messenger is a highly efficient yet gentle reagent that is perfectly suited for applications such as gene expression, CRISPR, and cell reprogramming. Since there is no need for nuclear entry, mRNA transfection with Jet Messenger ensures quick protein expression that significantly outperforms leading competitor DNA reagents, as you can see here. Moving on now to one of our most versatile transfection reagents in the portfolio. Jet Prime is a superior reagent that allows for the co-transfection of plasmid DNA and silencing RNA. Providing exceptional transfection efficiency in a breadth of adherent cell types and significantly outperforming competitors, Jet Prime is your go-to reagent for a wide variety of applications, including plasmid transfection, gene silencing, co-transfection studies, and CRISPR-Cas9 mediated gene knockout. So the last reagent that I'd like to highlight today is interferon. This is a transfection solution that is perfectly suited for gene silencing studies with molecules such as siRNA, microRNA, as well as other oligonucleotides. With as little as 1 nanomolar siRNA, interferon is able to achieve over 90% gene silencing across a wide variety of both adherent and suspension cell types, ensuring fewer off-target effects and excellent cell viability compared to competitor reagents. Moreover, with the ability to deliver microRNAs and other oligonucleotides, you also get the option of using interferon to stimulate gene expression, as shown here on the right. And with that, I'd like to now bring you to my final slide. I hope this webinar has given you an insight into how Sartorius, together with Polyplus, is at the forefront of advancing cell and gene therapy through innovative transfection solutions. For more information on these products or on any of our solutions, please visit our website. If you would like to request a sample for one of our transfection reagents, please scan the QR code on the screen. Thank you for your time, and we look forward to hearing from you.
Thanks, Hina. That brings us to the end of the presentations today and to the end of the DDW Cell and Gene Therapy Showcase. I'd like to thank everyone for tuning in, and a big thanks to our panelists and presenters today. If you would like to listen to the webinar again, it will be available on demand at www.ddw-online.com. Thank you to the co-sponsors of today's showcase, Benchling, Sartorius, Applied Stem Cell, and Elegant Bio. You can now view Drug Discovery World content online on your mobile or tablet. Thank you for joining. Goodbye.